Okay, so now we're going to demonstrate meiosis. And in meiosis, this is the uh, production of germ cells or gametes, which is going to result in 4 sperm cells or 1 egg cell and 3 polar bodies. But we're going to start with a primordial, the parent cell. And so we're going to have chromosomes from mom, chromosome from dad. Right now we are going to be in interphase, and in interphase, during the synthesis part of interphase, we are going to duplicate our chromosomes. So we are going to duplicate these guys, and these should be so in interphase, we're just duplicating. When we get into prophase, which is the next step, is when these chromosomes continue to condense, and then we form, in prophase 1 of meiosis 1, we form homologous pairs. And homologous pairs are chromosomes from mom and dad that are about the same length. They're the same chromosomes, so they might be like chromosome 1 or chromosome 2, that have similar genetic information but not the same information. So because they're two completely different organisms. So what's going to happen is these guys are going to come together, and in prophase 1, this synapsis is going to form tetrads. And what's going to happen is there's going to be something called crossing over. And during crossing over, we're going to exchange some genetic information. So we're going to be taking some of these genes, that are in the same place on mom and dad. It's going to go bye-bye. And they're just going to kind of swap information. And what this does is this just ensures some genetic variation, and ensures that we're all unique. So, sorry, this is a bit of a mess, but this is the crossing over taking place, and this is prophase 1. And then what's going to happen is as this nuclear envelope is going to start to go away, and we're going to start bringing in some centrosomes with the spindles, and we're going to start to move into metaphase 1. And during metaphase 1, these homologous pairs are going to stay together, and they're going to line up in the middle of the cell on the metaphase plate, and then the spindles from the centrosomes are going to attach to the pairs. So it's going to attach to the chromosome, the whole chromosome. So attach. Sorry. And then we're going to move into anaphase 1. And during anaphase 1, these centrosomes are going to, and the spindles are going to start to pull these pairs, pull these chromosomes, to opposite sides of the cell. And the nuclear membranes are going to start to reform as we go towards telophase. And in telophase, these centrosomes and the spindles are going to go away. Oh my gosh, there's genes everywhere, which doesn't usually happen. The genes pretty much stay on the chromosomes, so just pretend that's not happening. And so telophase, the genetic information is going to go back into the nucleus. The nucleus is going to start to reform, and the chromatin is going to start to get a little bit looser. Cytokinesis starts to happen, where the cleavage furrow starts to form, and the cytoplasm divides. And then we finish with 2 daughter cells. And then these have each of these has the original number of chromosomes, but we're going to divide again because we want half that number of chromosomes spread between 4 daughter cells. So then we undergo meiosis 2, and we will be right back with that. Okay, so we are finished with telophase 1. And after telophase 1, we're left with 2 daughter cells that are genetically different. And from there, we go into meiosis 2. And in meiosis 2, we kind of bypass the interphase section and go directly to prophase 2. So prophase 2, that chromatin is condensing again. We're getting back into our chromatin form with our chromatids. And not too much going on exciting here. No homologous pairs are crossing over. We're just kind of, the chromosomes are hanging out. And you can see that we've got some genetically diverse chromosomes here. They've crossed over in the prophase 1. And then we move into metaphase 2. And then these chromosome pairs are going to line up again in the middle, but this time the centrosomes and the spindles are going to attach to the centromeres of each sister chromatid, and we are going to, oh my goodness, that one's kind of messed up. Anyway, pretend there's a spindle attached to this one. Yeah, so the spindles attached there, and then in anaphase 2, and then this, again, this nuclear membrane kind of goes away. I'm going to leave this there to show the division of the cells. So in anaphase 2, we're pulling these chromatids apart. We're pulling those to the opposite ends of the cells. And so then we have, we start to move towards telophase, where we start to develop the nuclear membranes. The centrosomes go bye-bye, and the spindles, boop, boop, boop. Bye, centrosomes. And now we are starting to come together here in telophase. Oh my goodness, my poor little genes. Yeah, so we're forming now 4 new nuclei, and cytokinesis is happening, and the cleavage furrow is dividing the cells. The cytoplasm is dividing. They're replicating their other organelles. And then at the end of cytokinesis, we have 4 haploid daughter cells that are all genetically unique. And in the case of sperm cells, we would have 4 sperm cells that are all genetically unique. But at the end of the meiosis 2 for an egg, we actually, these 3 become polar bodies, and this one big ova has all the nutrients and all the genetic information in that one big egg, and the rest of these guys kind of take a hike, get reabsorbed by the body, and that's the end result of oogenesis at the end of meiosis. So yeah, there you have it.